Hi everyone, my name is Stephanie Ira, and today we will explore the evolutionary advancement of our acquisition of 5 fingers. The sources I've used are the research paper Evolution of Hoxa-11 Regulation in Vertebrates is linked to the pentadactyl state from University of Montreal, and also How Evolution Has Equipped Our Hands with 5 Fingers from the website Science Daily. The links can be found on the page. So if you've ever wondered why we have 5 fingers, you are not alone. The researchers at the Institute of Clinical Research in Montreal, with Dr. Kamita, have discovered a part of this mystery. So it's been known for several years that the limbs of vertebrates, including our arms and legs, stem from fishes. The evolution that led to the appearance of these limbs, in particular the emergence of fingers in vertebrates, reflects a change in the body plan associated with a change of habit. As you can see in the picture, it is believed that these changes came from transitioning from an aquatic environment in water to a terrestrial environment, aka living on land. So how this evolution occurred is a fascinating question that goes all the way back to Charles Darwin's work, and it definitely didn't happen overnight. The fossil record indicates that our ancestors were polydactyl, meaning they had more than 5 fingers, which raises another question, which through what mechanism of change did this evolution favor the pentadactyly, which is our 5 fingers, among our current species. Just this summer, researchers found 2 genes, HOXA13 and HOXD13, that are linked and responsible for the formation of fins in our fingers. They state that this result is very exciting because it clearly establishes a molecular link between fins and fingers. So the discovery of these genes caught the attention of Dr. Kamita and her team, and they observed an important thing where they found that during development in mice and humans, the HOX11 and HOX13 genes are activated in separate domains of the limbs, but in fish, these genes are activated in overlapping domains of the fins. So they tried to understand the significance of this difference, what it meant, and what it meant for evolutionary lineages. So in the lab, they used fish-type regulation for the HOX11 gene, and they put that in mice, and the mice developed up to 7 digits per paw using the HOX11 gene, aka a return to ancestral status. Her team also discovered that the sequence of DNA responsible for the transition between fish and mouse type for the HOX11 gene. Dr. Kamita says, it suggests that this major morphological change did not occur through the acquisition of new genes, but by simply modifying their activities. She also stated that this reinforces the notion not only due to mutations in the genes and may come from mutations in sequences of DNA known as the regulatory sequences, as the regulatory sequences in this case are the ones that change the development between 5 versus 7 digits, not the actual genes themselves. So she found this big long summary in her research, but basically it comes down to the point that the evolution of HOX11 regulation has contributed to the transition from polydactyly in tetrapods to pentadactyly in existent tetrapods. And again, one more time, we see that the regulation has made the transition from 7 to 5, not different genes themselves. So the methods used in this experiment, they used polymerase chain reaction to duplicate the HOX11 gene that they wanted in zebrafish to knock in those alleles into the mice. So they were taking the regulation genes from the fish, putting them into the mice. Then they used staining, skeletal preparations, and imaging to get the right stains to get the images that you saw earlier. And then they used basic transfection and gene expression analysis to analyze these results, find the RNA, and find that it was in fact the HOX11 regulation RNA that was making all this happen. So in this example, the medium reporting was very accurate as it was from a science reporter. The hook that it had, though, it grabs you with having to answer the questions why we exactly have 5 fingers when the research doesn't explicitly state where that came from. And I think that is an appeal to non-science audience to kind of get them interested in something they wouldn't normally be interested in. And we're going to go over some constraints or future implications. So Dr. Kamita did state at the end of her report that at present, technical constraints do not allow for identifying this type of mutation directly in patients, hence the importance of basic research using animal models. So for ethical reasons, we can't directly do this on humans yet, but maybe in the future there'd be an ability to change these genes, be able to make 7-fingered people, maybe webbed, make little mer babies or things like that. But at the moment, there are none.